In this video, we'll learn how Mendel learned about heredity by conducting his experiments. So, what do parents give their offspring that confers or transfers similarity? It wasn't until the mid 1800s that any real headway was made on answering this particular question. And it was at that time that Gregor Mendel, this monk that was living in what is now the Czech Republic, conducted these studies that not only shed light on this question, but practically answered it completely. Mendel understood the essence of the genetics puzzle and set out to piece it all together. When Mendel turned to questions about heredity, there were no obvious answers. In fact, one idea that had been really popular since the late 1600s suggested that an entire pre-made human, albeit a very little tiny one, was contained in every sperm cell. It's kind of funny to think about now, given what we know. But though this was very imaginative, the idea just couldn't explain why children resemble both their moms and their dads, not just their dads. Another popular idea suggested that offspring reflect a simple blending of their two parents' traits by blending of the blood. But how then could brown-eyed parents give birth to blue-eyed children? Sometimes scientific breakthroughs are made because a new technique is invented or a lucky observation is made by chance. Neither one of these, though, played a role in Mendel's success. He didn't do anything radically new, but he simply applied the tried and true process of methodical experimentation and scientific thinking, what we call the scientific method. In particular, three features of Mendel's research were critical to his success. The first feature is that he chose a really good organism to study. That's really important. He chose the right organism to study. He chose the garden pea. And it's not that Mendel had a particular fondness for vegetables. In fact, his goals were to understand inheritance in all organisms, not just plants. But cats and dogs, or even mice, wouldn't have served his purposes very well because they would have had to have been too hard to take care of in large numbers, in the large numbers that he required, especially. He would have needed thousands and thousands of individuals. Humans, too, would have been a really terrible study organism because we take far too long to breed and won't produce any offspring on command. Pea plants, on the other hand, were simply the right tool. They're relatively easy to fertilize, and you can do this manually by a process called pollen dusting. A single cross, the process in which male pollen is used to fertilize eggs, produces numerous offspring. And in addition, pea plants reproduce quickly, so Mendel could conduct experiments that included multiple generations. The second feature of Mendel's research that led to his success was that he chose to focus on easily categorized traits like shape and color. For instance, all peas of the variety that Mendel studied are either round or wrinkled in shape, with nothing in between. In addition, all peas are either yellow or green in color, never any intermediate shade. In all, Mendel looked at 7 different traits of the pea plants, but for each trait, only 2 variants ever appeared. Mendel and his research assistants could easily observe and unambiguously identify the trait variants. And lastly, to ensure his success, Mendel began his studies by first repeatedly breeding together similar plants until he had many distinct populations, each of which was unvarying for a particular trait. He described these plants as true breeding for that trait because they always produced offspring with the same variant of the trait as the parents. For example, when true breeding round pea plants were crossed together, they always produced plants with round peas. True breeding purple-flowered pea plants always produced purple-flowered offspring, and true breeding white-flowered pea plants always produced white-flowered offspring. It took a lot of prep work to establish these populations, but once Mendel had them, he was in a position to set up all of the different crosses that enabled him to piece together the genetics puzzle. So now that he had obtained his groups of true breeding plants, Mendel began a straightforward process of experimentation. He crossed plants with different traits. For example, he crossed plants with green peas and plants with yellow peas, and observed large numbers of their offspring. counting the number of plants that produced green peas and the number of plants that produced yellow peas. Please, yellow peas. And this cross we write as green X yellow cross. And this always resulted in plants that produced only yellow peas. So then Mendel from that devised a hypothesis that would explain his observations and generate predictions about the outcomes of further crosses. Then he conducted those crosses to see whether the predictions generated by his hypothesis would bear out. There really was a simple elegance to Mendel's work. From his predictions, he articulated simple scientific questions, if you remember that from the scientific method we learned about at the beginning of the year. He was able to articulate these simple scientific questions that could be answered by his experiments, which is the requirement for scientific questions to be scientific. An example of one of his questions would be, if I cross a plant that produces wrinkled peas with a plant that produces smooth peas, will the offspring plants produce wrinkled or smooth peas? His crosses would come out either one way or the other. Everything was very black and white. This process of performing well thought out, rigorous experiments was a radical innovation for biology. In the next section, we're going to explore his results and see that his results allowed Mendel to clearly and definitively describe how the traits he studied were inherited.